Hi everyone, welcome to Chapter 16. This is all about gene expression. So, you know that every cell in your body contains the same DNA, except a few exceptions like mature red blood cells. But every somatic cell in your body has copies of the same 46 chromosomes, and therefore, that means that every cell in your body has copies of the same roughly 30,000 genes. So if every cell contains the same genes for instance, eye color, hair color, height, muscle fiber, contraction, speed, metabolism, 5 fingers on each hand, how is it then that muscle cells are different from nerve cells if they contain the same genes? How is it that your heart is functionally different from your liver? And how is it that your mother really does not have eyes in the back of her head? Well, the fact is only certain cells will express the genes for eye color. And those cells will not express the genes for blood filtration. And neither of those will express the genes for muscle density. So the point here is not all genes encoded in your DNA will be transcribed and subsequently translated into proteins by every cell in your body, because each cell is specific for various proteins, even though each cell has the potential for making all of these different types of proteins. So the expression of specific genes is highly regulated, and there are many levels of control here. So this ensures the proper expression in the proper cell at the proper time of the proper protein. And so when that process goes awry, and it does from time to time, bad things can happen. You can see from the illustration here that we have a leg growing where there should be no leg, right? There should only be 2 hind limbs for this poor frog, but we have 3 hind limbs. So things happen, mutations happen, things go awry. But when everything goes as it should, you don't have an eye where your ear should be, right? You have ears where your ears should be. And that's all part of gene expression, or gene regulation rather. So gene expression, phenotypic expression, is based on the regulation of the genotype, and that's what this chapter is all about. So your objectives here are explain why every cell doesn't express every gene all of the time, compare and contrast prokaryotic and eukaryotic post-translational modification. There are some variations there. Explain the roles of activators, inducers, and repressors in gene regulation. You should be able to tell just from the terms what they actually do. Describe eukaryotic epigenetic gene regulation. Discuss the role of transcription factors in gene regulation. Describe how the initiation complex controls translation. And describe how changes to gene expression can cause cancer. So gene expression is the process of turning on a gene to produce a messenger RNA and subsequently have that translated into a protein. Now each cell controls when and how its genes are expressed. So the control of gene expression conserves energy and space. So in other words, transcription and translation of RNA, those are extremely high energy processes. They cost a lot of energy. And your body does not want to waste energy. It doesn't want to expend energy where it doesn't have to. Part of homeostasis is to use energy only when you need it, to conserve it when at all possible. So it's energetically costly to do those processes all the time in every cell, and if every cell expressed every protein all of the time, if all of your muscle cells expressed the genes to make eyeballs, those cells would have to be ginormous. So by necessity, gene expression is highly regulated. So malfunctions do occur, and those malfunctions can lead to unwanted phenotypic changes, including cancer. So let's start out with prokaryotic gene regulation. Okay, you know that prokaryotes like bacteria are much simpler than eukaryotes like humans, and so everything for a prokaryote is at a smaller scale and a much simpler process than it is in eukaryotes for obvious reasons. But because prokaryotes, like bacteria, my example here is E. coli, are structurally much simpler and functionally less complex than eukaryotes, like humans, gene regulation in prokaryotes is certainly less complicated. So recall that transcription and translation occur simultaneously in prokaryotic cells. Remember that as the DNA in bacteria, E. coli for instance, as the DNA is being transcribed, then RNA polymerase can jump right on the transcription and start translating that transcription. And so even before the transcription gets completely transcribed, proteins are being translated off that transcription, and they will continue to occur, right? So when the protein is no longer needed by E. coli, for instance, transcription of the genes cease. That's the primary way that prokaryotes, like bacteria, control gene expression. So regulation of gene expression in prokaryotes occurs at the level of transcription. Easy enough, yes. And so here on the left we see the bacterial cell. There's a plasma membrane. And here's the single circular DNA chromosome, right? Single circular chromosome for bacteria. And so here you see the DNA being transcribed. There's the transcription. And even before it is completely transcribed, the ribosomes are jumping on there and they are translating into protein. They're translating those proteins. And then when the cell no longer has a need for a particular protein from this transcription, then translation will stop. And ultimately transcription will stop. It's a little more complicated in eukaryotes, as we see over here, and I'll reference this illustration again in a second. So in eukaryotic gene expression or gene regulation, okay, so here is my simplified version of the central dogma, right? DNA gets transcribed into RNA, and RNA gets translated into protein. And my numbers here represent the places in this process where gene regulation can occur, and there are 5 of them, namely epigenetic regulation that occurs in the nucleus after the DNA is uncoiled, or even before. If the cell doesn't want to transcribe a particular protein, then it just leaves the DNA wrapped up around histones and coiled together in nucleosomes, packs of nucleosomes. The DNA on the inside of a nucleosome, polymerase cannot get to that DNA, and so if polymerase can't get to it, then those genes cannot be transcribed and ultimately translated. So that's one area of gene regulation. That's the epigenetic area. 2 is in transcription. So we can regulate, or the cell can regulate transcription, can regulate genes at the level of transcription during the actual process of transcription. We'll see it in a little bit. Post-transcriptional regulation can occur. That's in the cytoplasm after transcription, right? After we make the we get the primary transcription and we process it and we send it out of the nucleus into the cytoplasm, we can do some regulation there. We can make some adjustments there to control regulation or ultimately translation of proteins. We call that post-transcriptional because it's after transcription. Then there is translational control or translational regulation that occurs during the actual translation of proteins. And then there's post-translational, after the protein has been translated. Post-translational, we have some control over it there as well. Okay, so here's a quick check for you. At which level does regulation of gene expression in prokaryotes occur? Your choices are post-translation, translation, epigenetic, and transcription. And if you answered at transcription, then you're absolutely correct. Remember, we only have post-translation, translation, epigenetic control or regulation in eukaryotes. So only at the transcription level do prokaryotes regulate gene expression. Okay, now with regard to prokaryotes, we have a couple of terms here. First of all, there's this term operon. Now, an operon is a suite of genes. It's a series of genes. It can be 3 or 4 or 10. Right? We don't care how many. It's just a bunch of genes that are encoded together. They get translated together and they form a protein that works together to do a thing, to do a job. We call that an operon. It's a suite of genes encoded together. They're translated into a block of proteins which work together to control or to regulate a single biochemical pathway. Remember that term. We have a repressor which prevents transcription of a gene in response to an external stimulus. Okay, so this is a protein which represses transcription. Okay, it stops or hinders transcription. It represses it. An example, and the two examples that I'm going to show you here and that are in every basic biology textbook that has ever been printed in the 20th and 21st centuries. One is the TRP operon. Now these repressors are operons, so it's a bunch of genes, but it's TRP. TRP stands for tryptophan. I'll explain that in a minute. Then you have the activator, which is an operon that increases transcription, so it activates transcription in response to an external stimulus. And then you have an inducer. An inducer is an operon that can go either way. It either represses or it activates. So you have repressors, which are operons that prevent transcription. You have activators, which are operons that enhance or increase transcription, and then you have inducers. They can induce transcription one way or the other. They can slow it down, they can speed it up, they can stop it, they can make it go, depending on the needs of the cell. And the one inducer that we'll talk about here is the LAC operon, or the LAC operon, and that stands for lactase or lactose. All right. So let's go to the video. Okay, Nicole Lance, I like her videos. Simple explanation of the TRP operon. Okay, remember this is a repressor. TRP stands for tryptophan. Watch the video and then come back. Okay, so from the video, this is a screenshot from the video. Nicole explains the TRP operon very simply and very well, I think. But so this is an image that you saw in the video if you watch the video. Okay. So we have here's the DNA and here are the genes. These are the tryptophan genes. So this TRP, E, D, C, B, and A. You don't have to remember all this, but when these genes are translated, they form proteins that together make tryptophan, and that's what you see down here in these little green dots. Okay, these are tryptophan. Now, when the operon is working, okay, when the operon is not there, I should say, when the operon is not in place, then the RNA polymerase can jump on here at the promoter, right, and it can go through and it can translate these genes. And these genes will join together and form tryptophan, one of your amino acids. Now, if there is enough tryptophan, okay, so tryptophan, the bacterium needs tryptophan in order to survive, okay? Let's just say that. And we don't need to know details of what, but it's like oxygen for us. We need it. We have to have it. Okay, so when tryptophan is needed, then RNA polymerase jumps on there, translate the genes, and tryptophan is made. Now, if there is enough tryptophan in the cell, then it's going to be a waste of time and a waste of money. In other words, a waste of resources, energy, to make more tryptophan. So if tryptophan is plentiful, here's the key. If tryptophan is plentiful, then tryptophan, okay, the operon here in this orange box is, just think of it as down here somewhere. But when tryptophan is plentiful, there's no need for the cell to make more tryptophan, so tryptophan will bind to the operon. That changes the configuration of the operon, and the operon is able then, only when tryptophan is attached, to bind to the operator. And when the operator is blocked, then RNA polymerase cannot go beyond that. Okay, RNA polymerase cannot hook up with the message, and therefore it cannot further translate these genes, which means that no more tryptophan will be made for the moment. Okay, does that make sense? So look, when there is excess tryptophan, that's what you see here. Tryptophan is a co-repressor. Tryptophan binds to the repressor to make it active. Now when the repressor is active, then it binds to the operator and it blocks RNA polymerase. Does that make sense? So when the operon is repressed, it binds to the operator. When the TRP operon is repressed, right, it's a repressor, it's going to stop or hinder transcription and translation, or transcription here, and ultimately down the road translation, right? It's going to repress that. So when the operon is repressed by tryptophan, it's going to block the operator here, and these genes cannot be transcribed and translated. So the operon prevents TRP gene expression. Okay, it represses gene expression. And then look, when tryptophan runs low, then the tryptophan will pop off, the operon will pop off, RNA polymerase will pop on, and the genes will be translated, and more tryptophan will be made. Okay. Quick check. So when tryptophan is plentiful, the TRP operon is repressed, thereby preventing the expression of the TRP genes. Now this is a true or false question. So when tryptophan is plentiful, the TRP operon is repressed, thereby preventing the expression of the TRP genes. So remember from the image that I just showed you, when tryptophan is plentiful, tryptophan binds to the repressor binds to the operator, and that blocks the further expression of these genes. Okay? That's definitely true. All right, let's go to the video here. Once again, Nicole Lance explains the lac operon. Remember, lac operon can exert both positive and negative control. Okay, so watch the video and we'll come back here. Okay, so remember that the lac operon, now lac stands for lactose. Okay, so here we're talking about lactose. So all organisms on the face of the planet prefer glucose as an energy source. However, some bacteria, when glucose is not plentiful in the environment, some bacteria can feed off of, can make energy from lactose, which is another sugar, right? That's milk sugar. So some bacteria can use glucose, preferably, that's number one, but when glucose is in short supply, then they can turn around and use lactose. And that's what the lac operon is all about, okay? So the lac operon, once again, it's a suite of genes, lac Z, lac Y, lac A, that code for various proteins. And those proteins join together to make lactose, which is that milk sugar. Now, for expression of the lac operon to occur, the repressor must be deactivated in the presence of lactose. So normally, here is the operon, and normally it's going to be right here at the promoter. Okay, blocking the translation of the lac genes, right? Because in other words, there's lots of glucose there. We don't need to, as the bacterium doesn't need to feed off of lactose. We have plenty of glucose, and so we don't need to make lactose. That's the deal. Okay, so however, when lactose becomes plentiful, or lactase becomes plentiful, see, then we need a way to digest lactose. Okay, so when glucose becomes scarce and lactose becomes plentiful, so lactose in the form of allolactose, you don't need to remember that, binds to the repressor to inactivate the repressor. Okay. And then this other little pair of proteins here, this CRP-CAMP, that's cyclic AMP. You don't have to remember that, but it binds to the promoter, and it just enhances the binding of RNA polymerase. Okay, so normally this repressor is up here blocking the promoter, connected to the operator, blocking the binding of RNA polymerase. Now, in the presence of lactose, no glucose, presence of lactose, lactose binds here, right? This is normally up here. Lactose binds to the repressor, changes the configuration, the repressor drops off. This little enhancer comes in here, binds to promoter just to enhance the binding of RNA polymerase. RNA polymerase can then translate these genes, make proteins, okay, and those proteins are going to make lactase in order to digest the lactose. All right, that's how that works. Okay, the lac operon exerts both positive and negative control, and here's how that happens. As a negative regulator, it's a repressor, and so when active, it prevents. Okay, so when it's active, it's connected to the operator. And when that happens, RNA polymerase cannot bind and cannot make those lac genes. So that's the negative control. As a positive regulator, remember, lactose binds here, pulls this off, and this little cyclic AMP pair binds to the operator just to enhance binding of RNA polymerase. Don't remember that detail, but I'm just telling you why that is here. But as a positive regulator, as the repressor comes off, right, then when this attaches to the promoter, it supports RNA polymerase binding. So this can either negatively regulate the lac operon by binding, or it can positively regulate the lac operon by coming off, right? So it can do one of those two things. Now, with regard to eukaryotic gene expression, here we see on the right side, this is a nuclear envelope, of course. You can see little pores there. And remember that transcription takes place in the nucleus, inside of the nucleus. And so here you see this is the DNA strand, and here you see it says pre-messenger RNA. This is the raw transcript, okay? This is the primary transcript. And then remember, we have to process it first, and then it can leave the nucleus and get translated. So we have to process it into the mature RNA, that's what you see here, and then it goes through the pore into the cytoplasm, and then the ribosomes can jump onto the message and start translating those proteins. So where all of that occurs inside of the cell and is happening simultaneously inside of the prokaryotic cell, we have all of these different steps that must occur, right, before we get protein out the other end. And so the point here in showing you this is to let you know that regulation for eukaryotes can occur here, 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 all right, and here. So there's 5 different places where regulation can occur. Look at those individually. So epigenetic regulation involves the packaging of DNA within the nucleus. And you know now that DNA is wrapped around these proteins called histones, and you get a bunch of histones together with their associated DNA, and we call that a nucleosome. And then nucleosomes are wrapped together or bound together in these big packages inside of the nucleus, and that's how you get all of that stuff. Remember in that little tube that I showed you of my DNA? Yeah, that's how you get all of that stuff that you can see with the naked eye into microscopic cells. It's the packaging process. Okay, so the point here is within the packaging, all right, this DNA, which is genes, that is wrapped up within these bundles of nucleosomes and histones, DNA polymerase cannot, or RNA polymerase cannot get to those genes to transcribe those genes, okay? All right? So all this DNA here is inaccessible. Okay? That's where regulation of transcription of those genes can take place. And if you need those genes, right, if the cell needs those genes, then these things will unwrap and unravel, and the DNA will become accessible to DNA polymerase. All right, so that's epigenetic regulation. Transcriptional regulation is during transcription, right? So like transcription in prokaryotes, eukaryotic transcription requires binding of RNA polymerase to the DNA template for you Southerners in the house. And because RNA polymerase cannot initiate transcription in eukaryotic cells, other proteins, we call them transcription factors, must prepare the DNA for RNA to bind, okay? RNA cannot. Okay, so remember those single-stranded binding proteins, and remember all those things we talked about before with regard to transcription in chapter 15. Okay, all those things must happen to prepare the DNA to be transcribed and translated. All right, so we have promoters. These are regions upstream of the coding sequence. Site of transcription factor binding promotes transcription by RNA polymerase. Okay, promoter, it promotes transcription. It promotes binding of RNA polymerase, thereby promoting transcription. We call it the promoter. The TATA box, we call it the TATA box. It's a sequence within the promoter. So the promoter promotes transcription by RNA polymerase. And the way that RNA polymerase knows where to go is because of this TATA sequence. We call it the TATA box. It's a sequence within the promoter that binds transcription factors, binds those transcription factors. And it's the place that RNA can recognize and say, okay, I can bind here and initiate transcription. And that's what happens. It's called the TATA box. It's part of the promoter sequence. Okay, so regulation of transcription. Here's a video from the Khan Academy talking about regulation of transcription. See you in a bit. Oh, and then here's Professor Dave explaining regulation of gene expression. He talks about operons, epigenetic regulation, and transcription factors there. Okay, so we have epigenetic regulation, we have translational regulation, transcriptional regulation, and now we have regulation after transcription, post-transcription. Okay, so remember that the eukaryotic transcripts must be modified before they can be shipped out of the nucleus. We have to get rid of the introns, we have to put on a 5' cap and a 3' poly A tail. So we have to do all of those things. So in each one of those processes, regulation of genes can occur. Okay. So here is a primary transcript up here. We're going to remove these introns. Okay. And so now you have gene 1, gene 2, gene 3, and gene 4 all separated by introns. Now when you remove those introns, we have to spliceosomes, right, come in and they're going to ligate those genes together. But they can tie those genes together in different ways. So as you can see here, here's gene 1, gene 2, and gene 3 spliced together. And over here is a different scenario. Here's gene 1, gene 3, and gene 4 spliced together. And when those genes are spliced together, then they're going to be translated into whole different proteins. And so part of the post-transcriptional regulation involves removing introns and then splicing these genes together in a myriad of ways. And each time you splice together, a different combination of genes, you get a different protein. So that's another way to regulate gene translation and protein production, and ultimately phenotypic expression after transcription. Okay. Here's the second part. So in addition to removing the introns, right, we have to add the 5' cap and the 3' poly A tail to the message. Again, now this is before it leaves the nucleus to get translated. So we're still post-transcriptional. All right, so down here you see the message. We removed the the introns. We've spliced together, we've ligated together the exons, and now we need to put on the 5' cap and the 3' tail. During the process of adding the cap and the tail, you can the cell can manipulate things even before that process, so that it can regulate gene expression at the process of modification. All right, that's part of post-transcriptional regulation as well. I'm not giving you a lot of details here because it's not necessary. You just need to understand that this is another area of regulation, post-transcription. And then once you get the mature message, then it can be shipped out of the nucleus into the cytoplasm to be translated. So this is translational regulations. Or translation begins when an initiation complex binds to the small ribosomal subunit. Remember, the ribosome is a small unit and a large unit, and the initiation complex, here we see the eukaryotic initiation factor 2 right there, binding to the ribosomal 40S subunit. Don't worry about the details. All right, so when the eukaryotic initiation factor is phosphorylated, as you see there's a little phosphate group, then the large ribosomal subunit cannot bind and translation cannot commence. So the small subunit, ribosomal subunit, has to first bind to the large ribosomal subunit, because remember, it's the large subunit that has the three, right, the E, P, and the A sites in it to receive the transfer RNAs. It's the large subunit that has those sites in it. So until those sites are present, translation cannot occur. And as long as EIF2 is phosphorylated, that large subunit cannot bind to the small subunit. And when those sites are not there, translation cannot commence. We get rid of the phosphate, then the small ribosomal subunit can receive the large subunit with the three sites in it, and when that happens, then translation can begin. So there's regulation that can occur at the level of translation. And then in post-translation, okay, so chemical modifications, proteins may result from external stimuli, including stress, malnutrition, heat, and UV exposure, okay? So after the protein gets translated, then things can happen, external things can happen. Stress, heat, overexposure to ultraviolet radiation, all these kinds of things can affect the protein. And when those proteins are affected, right, the phenotype is affected, then the cell has mechanisms to come in and dissolve, if you will, or break up or tear apart that protein. Now, proteins, as you know, are made up of long chains of amino acids, and your cell doesn't want to waste anything. So if you have a protein and it gets degraded, it gets broken apart, it gets digested, if you will, then it's going to be broken down into its constituent amino acids, which then can recirculate and be used again in making another protein. So modifications can result due to these external sources, and your cell, by marking damaged proteins or unneeded proteins, the cell can eliminate those proteins. So here's a protein that's either damaged from UV exposure or malnutrition or whatever, or it's just excess protein, it's not needed, it's not going to be used by the cell. Then here's our old friend ATP. We can phosphorylate the protein, adding a ubiquitin. Ubiquitin, that's like a little flashing light that says, over here, this protein is no longer needed, get rid of it, right? And so a proteasome, which is another enzyme, will come over here, wrap around this protein, with its red light flashing, and we'll tear it apart, we'll break it apart, we'll digest it, and release its constituent amino acid residues. These amino acid residues, there's an MET and an ASP and a PRO and so on and so forth in there. And those amino acids can be reused by the cell to make a new protein. All right. So even after translation of the protein, if something goes awry or if it's just excessive protein and your cell doesn't need it anymore, we can degrade those proteins, release their constituent amino acid residues, and reuse them. So even after translation, your cell has some control over phenotypic regulation. Okay, let's talk briefly about cancer. So remember, here is the cell cycle. And remember, in the cell cycle, there are various steps or various points of regulation where checkpoints, right, where the cell checks itself, and if everything is going swell, then the cell cycle can proceed to the next section, right? So there's several checkpoints in the cell cycle, and if something goes awry here, then that cell can be destroyed or degraded or dissolved, right? And the process will not continue, so that cell will die. So various proteins monitor these checkpoints, the three checkpoints that you know, G1, G2, and M. And if mutations are there, then these proteins will stop the cycle. However, sometimes mutations slip through, and if mutations are allowed to slip through, damaged cells can then multiply. And when damaged cells multiply, you get it's going to continue forever, right? Forever and a day. Damaged cells are going to continue to multiply if you don't get rid of them in the very beginning. And so cancer itself is the uncontrolled duplication and growth of damaged somatic cells. So when the cell does not successfully check itself at one of these checkpoints, and then these cells that are damaged go into mitosis, and they get duplicated and duplicated again and duplicated again, and every duplication, successive duplication, is a damaged cell. Uncontrolled duplication and growth of damaged cells, that's cancer. So cancer is a disease of altered gene expression. Remember that proteins can be turned on or turned off as a result of dramatic alteration of the activity of the cell. So a gene that is normally not expressed in a particular cell can be turned on and or expressed at a higher than normal level. And that altered gene expression, which now is a definition of cancer, altered gene expression can occur at any level, at any of these 5 levels that we've talked about before, right? The seeds of cancer can occur. So we have tumor suppressor genes. Now by their name you can tell what their job is. Their job is to suppress tumors. Tumor suppressor genes. Their job is to prevent inappropriate excessive cell growth. The most well-studied tumor suppressor gene is P53. That's something you should remember. P53 is a transcription factor that binds to promoters to initiate transcription. Now, if P53 is normal and working okay, it's going to bind to the promoter, initiate transcription. Transcription is going to occur, translation will occur, and those nice, wonderful, healthy cells will be, those nice, healthy genes will be translated, and you'll get nice, healthy proteins. However, if something goes wrong with P53, right, then bad things can happen. As you can see, the stat here says that P53 is mutated in more than 50% of all cancers. So in more than 50% of all cancers where P53 has been looked at, P53 is mutated. So if P53 is working normal, everything works normal, and you get normal phenotype. If P53 is mutated, then in 50% of those cancers, 50% of those situations, you end up with cancer. Now, proto-oncogenes are cell cycle regulators, and when they are mutated, they become oncogenes. Onco means cancer, right? So these are genes that cause cancer. Proto-oncos, these are pre-oncogenes. These are genes that can turn into cancer genes. All right, so proto-oncogenes can become, when they get mutated, become oncogenes and cause cancer. And one of the most well studied is the MYC proto-oncogene. MYC is a transcription factor, as is P53. Overexpression of MYC turns normal B cells. B cells, that's a type of immune system cell, white blood cell in the immune system, turns those B cells into cancer cells with uncontrolled growth. So MYC is a proto-oncogene, and when it gets mutated, it turns normal B cells into cancer cells. All right, so when it gets mutated, it becomes an oncogene and turns normal B cells into cancer cells. Okay, Vitae Studios produced this short little video, nice little illustration about tumor suppressor gene regulation in cancer cells. Take a gander. And here's a quick check. So some proteins like P53 act to prevent inappropriate excessive cell growth, which obviously can lead to cancer. That's what this section is all about. These proteins are the result of the translation of blank genes, proto-oncogenes, tumor suppressor genes, oncogenes, or alter genes. So you remember that P53 is a proto-oncogene, right? Oncogenes are cancer genes, tumor suppressor genes. P53 is a tumor suppressor gene, and it prevents the inappropriate excessive cell growth, which can lead to cancer. All right, so P53 is a tumor suppressor gene. Some proteins like P53, which is a tumor suppressor gene, suppresses the formation of cancer, right? That's how that works. All right, so epigenetic alterations. We're going to go through this real quick. I don't want you to worry about the details. Just know that cancer can happen at any of those stages, all right? So epigenetic is in the packaging of DNA. Silencing genes through epigenetic mechanisms is common in cancer. Cancer cells' DNA in the promoter region of silenced genes is methylated. Histone proteins that surround the region lack acetylation that is present when genes are expressed in normal cells. Don't remember the details. Combination of DNA methylation and histone deacetylation commonly found in cancer. That's the epigenetic modification leading to gene silencing. So in epigenetic regulation or alteration leading to cancer, the genes are silenced because they're wrapped up and they can't get unwrapped, right? Now, notice here it says these changes are temporary and can be reversed, interestingly. And so remember, this is before translation, before phenotype. Once you get to phenotype, it's much harder to do anything. That's when you have to do chemotherapy and you have to do radiation and you have to do surgeries. That's what you do on phenotypes. But at this point, nothing has been transcribed, nothing has been translated, so it's kind of easy at this point, it's kind of easy to knock out cancer or pre-cancer at this stage. And new drug therapies are being focused on reversing the nature of these processes, okay? Transcriptional control, alterations in cells that give rise to cancer can affect transcription. Mutations that activate transcription factors, such as increased phosphorylation, can increase the binding of transcription factor to the binding site in the promoter, and this can lead to increased transcription of that particular gene, which results in modified cell growth. So you can have an alteration or mutation at this stage, at the transcription stage, and what that will do, that will increase transcription. Increase transcription activation, which increases transcription of those particular genes. Uncontrolled transcription of those genes, which can ultimately potentially lead to cancer. Cancer post-transcriptional control. Here is stage 3. That's after transcription. Changes in post-transcriptional control of a gene can result in cancer. Specific cancers have altered expressions of miRNAs, and miRNAs can bind to 3' UTR of RNA molecules to degrade them. Too many miRNAs can dramatically decrease the RNA population, leading to decreased protein expression. And just from your textbook, several studies demonstrated a change in miRNA population in specific cancer types of specific cancers that have been studied. There has been a change in the miRNA population, and that happens at the post-transcription stage. Now, at the translational control and post-translational control, things are getting tough here, because once you get to protein, that's phenotype, and it's hard to make adjustments in phenotype. So translational or post-translational modifications of proteins can cause cancer as well. Modifications are found in cancer cells from increased translation of a protein to changes in protein phosphorylation to alternative splice variants of a protein, right? We looked at that before, where we remove the introns and then you can shift the genes around and splice them together in different combinations, and some of those combinations can be detrimental to the cell. So expression of an alternative form of protein can have dramatically different outcomes. Okay, here's an example. C-flip protein is a protein involved in mediating cell death pathway. Okay, comes in two forms: the long version, that's C-flip-L, and the short version, that's C-flip-S. Now, both forms are involved in initiating controlled cell death in normal cells. However, in colon cancer, for example, expression of the long form results in increased cell growth instead of cell death. So these two proteins, these two C-flip proteins, the long and the short, are both involved in cell death, in taking cells that are no longer needed and destroying them. However, it has been found in forms of colon cancer that expression of the long version, C flip L, results in increased cell growth instead of cell death. C flip L, C flip S work together for cell death, but when C flip L gets turned off, then it leads to increased cell growth. And of course, cancer, that's what cancer is, increased and uncontrolled cell growth. Okay, here's a quick check. The final one, where in the central dogma can adverse modifications leading to cancer occur? Translation, post-transcription, transcription, or post-translation? Well, the only one that's not mentioned here is epigenetic, and you know that it certainly can happen there. It can happen certainly in, so we have translation into protein, can happen there, as we just saw. Post-transcription, so we have epigenetic, transcription, post-transcription, that's where the RNA is being, the messenger RNA is being transcribed, so right after that. Before modifications, it can occur there. During transcription, it can occur there as introns are being removed and exons can be spliced together in different combinations. That can certainly lead to cancer. And post-translation, after the protein has been translated, can certainly occur there. So all of these answers are correct. Questions? Hit me up. And that's the end of Chapter 16. See you for Chapter 17 very soon.